Hello, everyone. Today, I am honored to introduce our latest publication in iMeta, entitled Arbuscular mycorrhizal fungal interactions bridge the support of root-associated microbiota for slope multifunctionality in an erosion-prone ecosystem. The Loess Plateau is a traditionally erosion-induced degraded region. Although it has been partially restored, there are still widespread low soil phosphorus availability. Slope is a key landscape feature and also the basic unit to evaluate the impact of landscape position in eroded ecosystems. Through the differentiation of vegetation and the redistribution of soil resources, slope position can strongly affect biological phosphorus utilization and the biodiversity ecosystem functioning relationship. Soil microbiota, especially the root-associated microbiota, plays a pivotal role in restoring degraded ecosystems by executing an array of essential functions. Rhizobia and arbuscular mycorrhizal fungi share a signaling pathway and mutually promote colonization in the rhizosphere, implying their coevolution that may largely contribute to phosphorus-limited ecosystems being restored with woody legumes, such as Robinia pseudoacacia. Unfortunately, existing knowledge about microbial diversity and functionality shifts along slopes is fragmented, with the focus limited to a single microbial kingdom or ecosystem function. Ecological networks serve as useful tools for comprehending complex biological interactions and can disentangle the roles of keystone microbial taxa in community assembly and ecosystem functioning. In eroded landscapes, dominant keystone taxa in network clusters have been identified as the engine to support multifunctionality and mitigate erosion impacts. However, microbial network complexity and interactions within and among fungal and bacterial kingdoms that are critical for maintaining multifunctionality during natural restoration processes are still unclear. In this study, we investigated the diversity and cross-kingdom biotic associations of root-associated microbiota, fungi, rhizobia, and bacteria, on a typical slope of an Robinia pseudoacacia plantation on the Loess Plateau. On this slope, we demarcated an area of 60 m x 30 m, with total 18 plots. To ascertain the support of bacterial and symbiotic kingdoms for ecosystem restoration, we explored their relationship with ecosystem functions across the slope, including individual functions, multidimensional functioning, and multifunctionality. We found that slope position significantly impacted multiple ecosystem functions and thereafter the average multifunctionality. 3 dimensions of slope multifunctionality were identified, jointly capturing 68.4% of the spatial variation in ecosystem functions. The first dimension, that was dominated by microbially driven carbon pools and organic matter decomposition, exhibited a distribution similar to that of the average multifunctionality. For root-associated microbiota, fungal richness and the composition of bacterial and rhizobial communities collectively drove slope multifunctionality. Soil nitrogen to phosphorus ratio emerged as a crucial abiotic factor influencing slope multifunctionality. Moreover, the quantification of RPOB gene revealed a marked amplification and selection of rhizobia within the rhizosphere, especially at the top of the slope. We also found that fungi can mediate the balance of microbial community assembly and stabilize cross-kingdom networks. Specifically, networks became more interconnected when fungal interactions were incorporated. 2 fungal ASVs were recognized as keystone species in cross-kingdom networks, assuming network connectors. The strength of positive associations was much higher than that of negative associations, aligning with the ratio of positive and negative edges. Biotic factors, especially those biotic associations between rhizobia and fungi, contributed much more than abiotic factors to bacterial and rhizobial assemblies and compositions. These findings underline how Robinia pseudoacacia enriches beneficial microbiota, forming an fungal-centered network to alleviate phosphorus limitation, which initiates a cascade of shifts including rhizobial symbioses. Such coevolution within the root holobiont suggests a tripartite partnership, with potential implications for slope multifunctionality under phosphorus limitation. Our findings confirm this viewpoint that less competition between rhizobia and fungi significantly supports slope multifunctionality, and their stronger positive association releases greater phosphorus mineralization potential. Furthermore, the symbiotic association among Robinia pseudoacacia, fungi, and rhizobia optimized microbial metabolism. Even after accounting for several abiotic factors, the support of fungal interactions for slope multifunctionality were robustly affirmed. Overall, this study reveals that fungal interactions are strongly linked to the delivery of slope multifunctionality. In response to resource limitations, Robinia pseudoacacia strategically recruits fungi, forming an fungal-centered network and establishing the tripartite association. We must highlight that this association is paramount to the successful restoration of degraded lands. However, a disproportionate number of woody legumes have been utilized. Minimally, these findings illuminate vital clues for rehabilitating degraded ecosystems and the indispensable biodiversity and services reliant on them. That's all, thank you.